Hello viewers, this is Carolyn Salome. Welcome to Tasty Anatomy online classes. Let's begin today's study with the clinical taster. I am presenting to you a case of a woman of 20 weeks gestation who has visited the hospital for her second transabdominal ultrasound. The imaging reveals less amniotic fluid, that is oligohydramnios, and this patient shows no evidence of amniotic fluid leakage. Less amniotic fluid, the case is, and no fluid leakage. So she is further referred to a high-risk perinatologist for a 2-week follow-up ultrasound. What could be the cause for the less amniotic fluid? Are you ready to investigate this cause and the case with me and then solve this? If so, you are at the right place with me to learn the developmental anatomy or the embryology of the urinary system or the urogenital system. Now this urogenital system consists of three components: urogenital and system, three words. In anatomy, the terminology system, we are well aware that it refers to a group of organs performing the same function, and uro refers to urinary. So uro refers to the urinary system and genital refers to the reproductive system. Now the embryology of this uro and genital and the reproductive system. Now we should have, or we should enlighten ourselves with a theme. The theme that lies in the development is number one, that the urinary system and the genital system, they are interconnected with each other. They are interconnected with each other. They develop from almost a common source. This is the first theme. The second theme is that the early components of the urinary system, the genital system takes up these early components for its development. That is the second theme. So, this theme has to be kept in mind while we study. Despite the common source of development, it is definitely worthwhile for us to consider the urinary system separately and the genital system separately, the embryology, to appreciate their individual anatomic role. Now, thus pushing us to form the learning objectives of this video, which is a two-part video, initially only about the urinary system, and this is part 1 video where we will be studying about part 1 development of the urinary system. Here the objective of this video is we are going to come across almost 10 to 12 new terminologies or the new structures which gives rise to the development of the urinary system. We are going to get to know about this, that is our objective. And going from the fact of known to unknown, the picture that you see on the slide is parts of the urinary system. A pair of kidney, a pair of ureter, a urinary bladder and the urethra. We are aware that the kidneys are posterior abdominal wall located and they are retroperitoneal, and the ureter is the passage of urine and the bladder is the temporary reservoir of urine, and the urethra anatomically it is different in male and female. Now these parts we are supposed to know how they are developed intrauterine. What structures helps to develop them? Now as you see here, there is kidney, and on the right side you see three names or three new terminologies: metanephros, metanephric blastema and the ureteric bud. These are the structures that gives rise to kidney. And the second part, the ureter, is developed from the structure called ureteric bud, while the urinary bladder is developing from vesico-urethral canal and another structure called mesonephric duct. Before we proceed, I'll just would like to recollect to you the word renal refers to the kidney and the word nephro refers to the nephrons or the kidney. That's why we come across again the terminology nephros, nephric, nephro. All these refers to the development of the kidney. The last structure, the urethra, as we saw anatomically, it is different in male and female. It almost develops from a common structure called urogenital sinus, which is shortly abbreviated as UGS. There are three parts, and these three parts variedly contribute to the male and the female urethra. All these, though we are going to get familiar with these terms, let's recollect once what are they. At the end of the study, ultimately you will be master of them, and you'll have a depth knowledge of how they develop and grow into different parts of the urinary system. Intermediate mesoderm is a new term for you. Nephrotome, nephrogenic cords, pronephros, mesonephros, metanephros, and the mesonephric duct, which is also called as the Wolffian duct, metanephric blastema. Follow the pronunciations with me so that you will be able to pronounce it for yourself. And ureteric bud, urogenital sinus, and last but not the least is the cloaca. These 10 terms, keep it in mind. Before we proceed, any embryogenesis, we should keep few things in our mind. Now, what is this embryogenesis? I'm sure you know the difference between embryo and fetus. If you don't know what is embryo and what is fetus, please take a note of it, search it for yourself, and comment it to me in the comment box. So there are 6 things that we should remember and observe and note while studying the organogenesis. First thing is the germ layer. There are 3 different germ layers. Which germ layer contributes to the development of the organ is must. Second is the timeline, that is which week it begins the organogenesis, when does it end, that is the timeline. The third one is the genetic factor or the genes that is inducing or signaling for the development, or the genes that can be mutated for the anomalies. We have to keep that in mind. The fourth one is the positional changes after formation, because every organ mostly they undergo some modification in the position and location. The next one that should be kept in mind is the vestigial part or the remnants, the remaining of the structures which stay back in adult life as a remnant without any function, that also has to be kept in mind. Beyond all this, the sixth point and the major point that has to be kept in mind while studying the development of organs is we cannot see intrauterine development, we cannot dissect them unless there is a factor called imagination present there. So I urge you to tap your imagination potentials and follow with me to the development of the urinary system. So in order to go, the first thing that have to be kept in mind, which germ layer. Now few of us must be not aware of this term germ layer. The germ layer itself is a Greek word which refers to budding, the cells from which the embryogenesis happen. First, a quick overview. This is the ovum and you have the sperm. Both of them they fuse, and that fusion is called fertilization, which gives rise to 2-cell layer. This further gives rise to 4-cell layer. Further divisions till the morula, and then you have 108 cell stage, a ball of cells which is tuft of cells are formed, and in between the cells there is fluid secretion taking place, which helps in pushing few cells at one end, the other cells lining the cavity which is formed within. Now this cavity is called blastocele, and the cells which line moved apart are called embryoblast, and the cells which line the cavity are called hypoblast. Let me tell you, the cells which are moved apart are called embryoblast. Embryo refers to the developing embryo. Blast refers the precursor cells. So the cells of embryoblast gives rise to the embryo. And these cells which are lining the cavity, they are below the embryoblast. Below means hypo, so they are called as hypoblast. Now this is the blastocele cavity. Now what happens, you have 2 different layer of cells within the blastocele cavity. Now keeping this in mind, let's see the germ layer. Again same thing in picturization, 2-cell layer for morula, and then the blastocyst and the blastocele formation taking place. Next, the same thing, what happens is, germ layer is continuing. what happens here in this picture, this ball of cell is going to get attached into the posterior wall of the fundus. Now this is the fundus of the uterus. These are the uterine tubes that extend. This is the body of the uterus. Here is the cervix. This is the cervical canal. This is the uterine canal. And this is the fundus. Here, this ball of the developing blastocyst gets attached here. Now, this part which contributes from the uterus, it forms the placenta. This one. And this part which is contributing here is the developing embryo. Same thing again, picturization. What you see here, this is the placenta, this part. This is the developing embryo which you see here, placenta connected by the cord. Now, so this results in further cavitation in the embryoblast cells. You can see the embryoblast cells, further there is cavity formed here. This cavity will be called as amniotic cavity, and cells are pushed on the top, they are called amnion, and the cells which are here, they will be called still as embryoblast and this as hypoblast. So there are bilaminar cells taking place, formation occurs. The top one is the embryoblast and the hypoblast with two cavities. What are the two cavities? The amniotic cavity and the exocoelomic cavity. Now the blastocyst seal is now called as the extracoelomic cavity. Now this further continuation is called gastrulation. The term gastrulation refers to development of three germ layers. Development of three germ layers is called gastrulation. Three germ layers. What is three germ layers? Just now we saw there is a top layer which is called embryoblast. Then we have a lower layer called hypoblast. Embryoblast, and the below one is called hypoblast. Now during development there is something which is formed here which is called the notochordal process, which forms this indentation. And after this notochordal process, this is also from the hypoblast, what happens is the cells from the epiblast migrate towards down underneath the epiblast and between the hypoblast, and they fill this area, giving rise to three layers of cells. Initially it was the embryoblast, the hypoblast. Now it gives rise to one which is called as ectoderm, two which is called as mesoderm, and three which is called as endoderm. Now formation of these three germ layer is called gastrulation. Now following that in picture, the same thing what we see here, you can see this video also. The epiblast cells, they are migrating down and they form the mesoderm, which is between the epiblast and the developing hypoblast as endoderm, and they fill the coelomic cavity. Now this results in gastrulation. This gastrulation follows by folding of the embryo. Folding of the embryo is resulting in the formation of body cavities. Now there is a plate of cells which has three layers. First layer, second layer, and the third layer. The first layer is the epiblast. The second layer is the mesoderm, and the third layer is the endoderm. Ectoderm, endoderm, and mesoderm. Now folding means this is the head end of the embryo, this is the tail end of the embryo. Now this embryo is going to undergo a head fold, a tailed fold and two lateral folds. We saw ball of cells, two layers were there. Now it becomes three layers. If we elongate and take a section, this is the ectoderm, this is the mesoderm, and this is the endoderm. It forms almost a disc like this. A plate like this is formed with three layers. Now after folding, what happens? This plate is now folded onto like this. At the head end there is a fold, at the tail end there is a fold, at the lateral region there is a fold, forming an embryo like this. Shaping at the head, tail end like this. This folding results in the formation of body cavities. Now you can see here in this video there is lateral folding forming in the body cavities. The mesoderms also, they relocate. The hindgut or the developing hindgut from the yolk sac is pinched inside and we can see the formation of the cavity here. Now this formation of the cavity results in the assortation of the mesoderm as three different ones. Lateral plate mesoderm, the middle one called as the paraxial, and the intermediate. This is of our concern, the intermediate mesoderm. Let's go on now to the genesis of urinary system. So we have come across this term. What is this term? Intermediate mesoderm. A very quick overview of what we have seen so far. A transverse section through the embryo, this is the head fold, this is the buccal membrane, this is the curvature, here is the allantois, here is the embryo. Now this is the head end of the embryo, this is the tail end of the embryo, this is the yolk sac of the embryo. Now at the head end and the tail end, what we see? So after folding, this is nothing but completely the ectoderm which is folded. This is the yolk sac and the midgut which is going to line inside, which forms the midgut, the gut tube. This is the gut tube. Next, we have the mesoderm in between. Now, if I am going to take a transverse section like this, if I am going to cut the embryo and view from top, I will see a cylinder. I will see a cylinder. This cylinder I will be having the developing, this is the outside is the ectoderm, and I will have the yolk sac being pinched inside the developing gut. On either side I will have the paraxial mesoderm, lateral plate mesoderm coming. This is the paraxial, this is the intermediate, and this is the lateral plate. Paraxial, intermediate, lateral plate mesoderm. Here I will have the neural tube. This is also the neural tube. These are the mesoderm layers and this is the endoderm layer. Now what will happen is this lateral plate also will fuse after the folding and it forms the extraembryonic coelomic cavity, and this is paraxial mesoderm. This is our concern, intermediate mesoderm. This is lateral plate mesoderm. Now the same structure, same thing I am going to show cut like this, and only this is the posterior half, this is the anterior half. I'm going to show the posterior part alone. I'm going to cut longitudinally, and I'm going to see only this part. If I'm going to see this part, this is the posterior abdominal wall, which is made of ectoderm. Here is the gut tube, which is developing. And here I have the neural tube. This is the developing aorta. This is the neural tube. And here I have the paraxial mesoderm, intermediate mesoderm, lateral plate mesoderm. Paraxial mesoderm will give rise to the somites. These are the mesoderms. This is the endoderm. Now, if this gut tube extends craniocaudally, this intermediate mesoderm also extends craniocaudally, and this fuses. This is what is happening, projecting. Now, this lateral plate mesoderm is going to undergo few changes and helps in the development of the kidney in this region, in the posterior. What are the changes which is happening? This lateral plate mesoderm is going to, now see this picture. This lateral plate mesoderm, now see here, this is the gut tube, the lower end is called cloaca, and this is the intermediate mesoderm that also joins into the cloaca, the tube of it. Now this is elevated. There is a ridge which is formed. The intermediate mesoderm forms a ridge, and this ridge is going to contribute for the development of urinary system and genital system. So I am going to call this ridge as urogenital ridge. So this is the urogenital ridge. This urogenital ridge is going to extend from the head region to the tail region. It is going to extend from the head region to the tail region, and that we call it as a rounded structure called cord, which is going to develop into the kidney. So hence it is called development of the nephron. So nephrogenic cord. This nephrogenic cord is unsegmented. Later it gets in 3 segments and it forms the kidney. Keeping this in mind, let's move on to see the picture. The urogenital ridge, which I mentioned, it is nothing but the elevation on either side of the aorta. Nephrogenic cord is the unsegmented column on either side of the aorta. You can see in the lower picture, it is projecting into the coelomic cavity. The lower picture is the transverse section. Next is nephrotomes. Nephrotomes are the segmentations of the nephrogenic cord. They are segmented as pronephros, mesonephros, and metanephros. I'll come back to this. All this now, second question is timeline, 4th week. The germ layer is intermediate mesoderm. The timeline is 4th week. Now this intermediate mesoderm gives rise to 3 segments. What are they? Pronephros, mesonephros, and metanephros. And on the side of this segment there are tubules arising, which is called mesonephric tubules. These are called mesonephric tubules also arise. So this pronephros, mesonephros, and metanephros are the segmentation craniocaudally which is happening. Urinary system develops from this. So first let's go on to study about the pronephros. Pro, the word refers to the initial kidney or the first kidney which is supposed to develop, but this development starts at the 4th week. There are almost 7 to 10 solid cells in the cervical region. Now what is the cervical region? If you see, this is the embryo, the head end of the embryo, this is the tail end of the embryo, this is the head region, this is the cervical region, this is the thoracic region, lumbar region and the sacral region. At the head's neck region, the pronephros starts beginning at the 4th week begins. But at the end of the 4th week again, they start regressing. All these pronephros start regressing before the mesonephros form. That's why the pronephros are called primitive kidneys and they do not serve any function. The mesonephros, that is the middle one, that is forming the till 8th week. It is a transient excretory function, but that also starts regressing. Now what happens here, you can see in this picture, this is the urogenital ridge which we were talking about. This is the gut tube. This is a part section projecting into the coelomic cavity. Now this mesonephros, there is a duct which is seen here. This is the mesonephric duct. You can see this is the mesonephric duct. From the duct there are tubules rising which takes the shape, elongated S shape, and the distal proximal end of it gets connection with the aorta, and tuft of glomerulus, capillary, all are formed and they drain into the mesonephric duct. All this takes place between 4th to 8th week, and the mesonephros also start functioning between. Here you can see the mesonephros. This is the pronephros which we were talking about. These are the mesonephric. You can see the S-shaped tubules also there. But they also start regressing because they are not going to form the definitive kidney. They form Bowman's capsule, everything, but they start regressing. Now the contribution of this formation of the mesonephros is done by a genetic factor called Wilms tumor suppressor gene. Keep this in mind, we'll come to it in a minute. Now as the ducts grow, all this takes place, the lower part of the mesonephric duct will go and join the ventral walls of the cloaca on 26th day. That's about the mesonephros. Next is the definitive kidney, which is formed by the metanephric kidney or the metanephros. All right. Now the metanephros, what happens to this metanephros? Just a quick overview. We have studied there is the metanephric tissue segmented as pronephros, mesonephros and caudally at the tail end the metanephros. Now this is regressing, the pronephros is regressing, the mesonephros is regressing, the metanephros is going to form the kidney, definitive kidney. Now on the side you have the tube which is running, into which the tubules were entering, the mesonephric tubules. This duct is called mesonephric duct. Now from the mesonephric duct, before it enters the cloaca, the hindgut, what happens? There is a small outpouching taking place. This outpouching is called ureteric bud. This projects into the metanephric tissue. Now this metanephric tissue, now it is called as metanephric mesenchyme or metanephric blastema. Now keep these two words fix it in your mind. Metanephric bud, megablastema and ureteric bud. These are the two forerunners in the development of the definitive kidney. The metanephros or form ureteric bud, as I told you, that is the sprouts on the day 28, and the metanephric mesoderm or the blastema gives rise to the mesenchymal cells, projects there. The ureteric bud projects here. Now what happens is the ureteric bud, as it projects, it contributes to the formation of the collecting system of the kidney, and the metanephric tissue contributes to the formation of excretory unit. We are going to find how. First thing, the ureteric bud penetrates the mesenchyme. Penetrates first. Second, the penetrating tissue, next it starts dilating. This is the ureteric bud. It projects into the metanephric mesenchyme. Now what happens? It starts dilating. This dilated part will form pelvis. They dilate craniocaudally. And they further divide into 20 generations, almost more than 20 generations, and the tubes, the second order enlarge and absorb the third order, and the fourth order forms minor calyces and the major calyces. So the ureteric bud contributes to the formation of the collecting system, whereas the excretory system is formed by the metanephric blastema. You can see a tuft of cells as a cap. It surrounds the tube. This cells undergoes elongation as S. They take the glomerulus, they take the blood supply, and they start differentiating as glomerulus. They elongate at PCT, Henle's loop and DCT, and finally they drain into the ureteric bud, and hence they contribute to the formation of the collecting system, the excretory system. So the two major resources of the metanephros or the definitive kidney is metanephric blastema, which contributes to the excretory unit, and the ureteric bud, which contributes to the formation of the collecting system. Now, the gene responsible for all of them, I told it's the Wilms tumor gene. Any faulty process in this gene results in the cancer of kidneys, usually for children under 5 years of age. It is also called as nephroblastema. You can see this in the picture. It is the remnants of the immature kidney. Now, the two key components, as we studied, the ureteric bud and the metanephric blastema, contributes to the definitive kidney. There is an interaction between these two tissues. If there is any faulty interactions, that can result in anomalies of the kidney. There is an anomaly which can happen is called renal agenesis. What is the kidneys may fail to develop either unilaterally or bilaterally. Now you can see in the picture, the first one shows agenesis, the second one aplasia, and the last one the hypoplasia. Now because of there is faulty interactions between the ureteric and the nephrogenic stromal mesenchyme, this can happen. Now, because of renal agenesis, there is less amniotic fluid. Now, why there is less amniotic fluid? Because renal agenesis causes dysplastic kidney. There is reduced fetal urine excretion, which results in the less amniotic fluid, which can result in a sequence of events called Potter's sequence. This baby will be at stillbirth or fatal after delivery. which can be seen. The symptoms are low set ears, big nose, pulmonary hypoplasia, respiratory failures, and limb deformities. We should always keep in mind, since uro and genital system go hand in hand, any faulty renal genesis can also result in faulty genital abnormalities as well. Now coming to the development, we are ending it. Now after development, we saw it is derived from the intermediate mesoderm. Next we saw the timeline, it begins at 4th week. Next we saw the gene which is responsible. The next thing which we should keep in mind is the positioning of the kidney and its relocation. Initially, the primordial permanent kidneys lie close to the pelvis, but due to the diminution in the curvature. Now, this is the curvature of the embryo. This curvature diminishes. It is well curved, then diminishes. The kidneys from the pelvic part, they start moving to the abdominal region. So there are three things which is happening due to the diminution of the body and the development of abdomen and pelvis. Now what are the three things? The kidney first moves cranially. Initially, both the kidneys are close to each other because they develop from the mesonephric duct which enters into the cloaca. But as they move cranially, what happened? They move apart from each other. This is the second happening of the positioning of the kidney. And the third thing, as they move apart, they undergo a medial rotation of 90 degree. Why? As the kidneys, they go up, they take the blood supply from the pelvic region. The hilum is ventrally placed. But as they move apart, the hilum rotates medially and there is 90 degree rotation taking place. These are the three positioning happening in the relocation and positioning of the kidney that takes place after the development. Now, during the early phases of migration, the metanephric kidneys regress, whereas the metanephric ducts are never regressing. They are retained because they are going to be closely associated with the development of the gonads. Now you can see the timeline of the ascensions of the kidney. Now if there is any faulty positioning and relocation that can result in anomalies, first what you can see, the first circle is the pelvic kidney if it fails to raise up, and second, if they are failing to move apart from each other and they can be fused in the midline forming a horseshoe shaped kidney, and this horseshoe shaped kidney can compress the posterior structures which is the abdominal aorta. Further, there can be abnormal renal arteries as well as they go. The function of the kidney, we see the kidney starts functioning, the definitive metanephros from the 12th week, but the urine is passed into the amniotic fluid. There is recycling, but it is not responsible for the excretion of waste because placenta is the one which serves the function. So here we end the completion of the formation of the kidney. This is the part 1 video of the urinary system development. I am going to show you a short video of how the urinary system starts developing. You can see the kidney develops from the intermediate mesoderm. The keyword, keep this in mind, the nephrogenic cords. They differentiate into pronephros, mesonephros and metanephros. The pronephros are the primitive ones. They start regressing. The mesonephros, the middle, and the metanephros in the lower part. Pronephros starts beginning at the starting of the 4th week, and by the end of the 4th week itself, they will start regressing. The mesonephros, they extend as into the mesonephric duct. They form elongated S-shaped tubules. They form the glomerulus. They form a temporary kidney, and they also start regressing by the week of 10. Then the mesonephric duct is still intact, which forms the gonads. Whereas the metanephric region, there is a small outpouching called as the urethric bud, which will start develop just at the junction of the duct, mesonephric duct into the cloaca. They project into the metanephric tissue, which is called as the metanephric blastema. Now these two, metanephric blastema and the urethric bud, they contribute to the development of the excretory part of the kidney and the collecting part of the kidney. The urethric bud contributes to the formation of the ureter, pelvis, major and minor calyces. As they progress, 20 further generations are formed and they form the collecting part of the urinary tract or the kidneys, whereas the nephrons are also further formed. The blastema or the metanephric mesenchyme, they form a cap around the duct, and this cap differentiates into a nephric vesicle initially, and this vesicle further forms tubule, and this tubule takes the shape of a S, and one end of the tubule gains the angiogenesis from the aorta, and it forms a tuft of capillaries, and then which designates itself as the Bowman's capsule, and the other end they elongate and differentiate, forming the proximal convoluted tubule, Henle's loop, and the distal convoluted tubule. Then the distal end goes and gets attached to the urethric bud part, the generation, and it forms the collecting duct. Hence, there are almost many millions of nephrons are also formed at the end of this stage. Now, there is also relocation. It is in the pelvic part. They start migrating up. As they migrate up, they also medially rotate and they move further apart also. The hilum, which was ventrally, now gets rotated medially. Now this is the end of the development of part 1 of the urinary system. I am going to now present to you back again the clinical taster which we saw, and come back to the case and try to solve it. Less amniotic fluid, no leakage. So what could be the cause? Renal agenesis. That is the suspected diagnosis of bilateral renal agenesis resulting in oligohydramnios. Thank you for staying with me and keeping these in mind. Now to try your understanding skill, I have placed a question. Please do read and place the answer in the comment box. During development of the urinary system, 3 systems form. What are they and what parts of each, if any, remain in the newborn? Now this answer in the comment box will help me to understand your understanding skill. Thank you for staying with me. Continue to stay with me to the rest part of the development as well. Thank you.